Bismillahirrahmanirrahim. 9700 A level bio. This is the AS level, and we're going to be discussing a paper 2, which is I've done one paper, and now this is the June 23 question paper 2-1. As you all know, this paper 2-1 or paper 2, any paper 2, is for 1 hour and 15 minutes. And everybody knows that you have the paper 2 the same very day this you have paper 5. So first you will do this for 1 hour 15 minutes. Then you probably have a break for about 10 minutes, and then you do another paper 5 for 1 hour and 15 minutes. And this is for 60 marks, while this paper 5 is for 30 marks. So now you have to realize why you've been given more time for 30 marks, because that's a lot of thinking paper, and it requires a lot of, to read the question twice and then of course start to answer it. Now coming to question number 1, figure 1.1 is a transmission electron micrograph of a cell from the stem of a pondweed, whatever Stoechenia pectinata. Magnification is into 2500, and you can say this electron micrograph because a light microscope maximum useful magnification is 1500. So it says state the evidence from the figure 1.1 that shows that the cell is from the stem of X pectinata and not from the mesophyll cell of a leaf. Mesophyll cell of a leaf will have a large central vacuole, and the cytoplasm is pushed to the periphery, and there'll be chloroplasts. But here, why is it from the stem? Because there are no chloroplasts. And there's a presence of small vacuoles. There's no large central vacuole. And the nucleus is very central. You can see this is the nucleus. Where is the nucleus? Let's see if you can label it. This is the nucleus. That's why I asked you to look at micrographs. And this would be the nucleolus inside it. So you must be able to pick this up, and you should Google it, electron micrographs of animal and plant cells, and look at least a few, 30, 40 of them to see how you can pick up the nucleus. So evidence that shows that the cell is from the stem, no chloroplast, presence of small vacuoles, or you can say absence of a large central vacuole. And the nucleus is central, or you can see the nucleus is not located at the edge or at the periphery. So this was a 1 mark question, and you have to give me any one of these answers. So this is how you would word it. So please have a look at it and see if you've understood it or not. Then part 2 of the question says complete each row in table 1.1 to identify a cell structure shown in figure 1.1 that carries out the function listed. Gas exchange. Now where is gas exchange going to occur? Gas exchange is going to occur in the cell membrane. Oxygen diffuses out, carbon dioxide diffuses in through the stomata, but then enters the air places and then through the cell membrane. So that could have been A, or it could have been E, because E is the mitochondrial membrane. So oxygen will enter the mitochondria for aerobic respiration. So it could have been A or E. Then production of subunits of ribosomes. This is the nucleolus. And the labeling of the nucleolus is C. Let's see it there. This is the nucleolus. Then active transport of ions would be, of course, channel proteins in the cell membrane. And this would be again A. You could have said A, you could have said F, which is the tonoplast, you could have said E, which is the mitochondrial membrane, and you could have said B, which is the nuclear envelope. Then aerobic respiration would be in the mitochondria. Mitochondrion or mitochondria, you can write either of them. And this was E. Then it says part B, plant vacuoles develop and vesicles fuse together. The vacuoles increase in size as more vesicles fuse. Always read the question very carefully and read it twice so that you understand what the question is asking you. Figure 1.2 shows the movement of vesicles within a plant cell during the development of a vacuole. Vesicles development of a vacuole, two different things. So you can see here is the cell surface membrane, then there's the rough endoplasmic reticulum, then you can see the multivesicular body, then key protein folded correctly, protein not folded correctly, and then you can see it, of course, these arrows going out of it, and these arrows coming here, from here, here, you can see this, and then you can see the vacuole increasing in size, and the Golgi body, and you can see the structures protein not folded. Now this is of course the ones here, protein not folded correctly are these ones here, these ones here, and then proteins folded correctly are these ones here, and these are the not folded correctly ones here again. So look at this very carefully, and then let's look at the question. Then name the process that is occurring at X. Where is X? X is this place. Now this is something taking in, so name this process. What is it? It's going to be endocytosis. Name the process that is occurring at S. You could have said endocytosis, but you cannot say phagocytosis. That would be a reject. That means a large number of students would have written that. Or you could have said pinocytosis, taking in liquid drinking, pinocytosis. Then it says some of the vesicles formed by the Golgi body pass to the vacuole. These vesicles contain proteins that do not have been folded correctly and some that have not folded into their correct shape. Some that have been folded correctly and some that have not folded into their correct shapes. Even I read it wrong. The proteins that have not folded correctly pass to the vacuole where they are broken down. Explain how proteins that have not folded correctly are broken down in the vacuole. And this is for 3 marks. So how would a protein be broken down? Easy. Hydrolysis. Use water to break the bonds. The enzyme would be a protease or a peptidase. And the peptide bond will be broken because the protein is made of amino acids and amino acids are joined by peptide bonds. So the peptide bonds will be broken to form amino acids. Very simple. Hydrolysis. Use water to break down the bonds. Either you said hydrolysis or you said use water to break the bonds. With the action of the enzyme protease, or you could have said peptidase, or you could have even said a proteolytic enzyme. Let's keep it simple, protease. Peptide bond broken to form amino acids. Hydrolysis, action of protease, peptide bonds broken to form amino acids. You could have even said to form peptides because they're actually, it's in 2 stages. One is to peptides and then it is to amino acids. But anything would have been correct. Then coming to the C part, small vacuoles in S. pectinata may have roles similar to lysosomes in animal cells. Describe the role of lysosomes in animal cells in defense against pathogens. Very simple. Formation of phagolysosomes, digestion of the pathogen, or you can say the bacteria or the microorganism. Then what are the enzymes required? You need the enzymes like proteases, lipase, carbohydrates, nuclease, or a lysozyme. And then what will be hydrolyzed will be either the polysaccharide or the peptidoglycan, which is the cell wall of the bacteria, or the proteins present in the bacteria, or the lipids in the bacteria, or the glycogen in the bacteria, or the DNA, which is made of nucleic acids, to form useful, or you can say to form soluble products. So you see the peptidoglycan would be broken up into amino acid peptide, glycogen, glucose to form useful soluble products. So you cannot use generalized words and say, breakdown of organelles. You remember bacteria does not have any organelle. Please remember that. They do not have any membrane bound organelles. Yes, they do have ribosomes, but they don't have any membrane bound organelles. Now please understand that the first mark scheme point is formation of phagolysosome. The other is digestion of the pathogen or the bacteria. Then by what? Digestion, how does the digestion take place? By enzymes. And you have to mention any two, either protease, lipase, carbohydrates, and nuclease. Because you see, what is the bacteria made about? It's a cell wall made of peptide or glycan. Cell membrane is a phospholipid bilayer. The cytoplasm contains many proteins which are enzymes. So all that needs to be digested. So hydrolysis of either a polysaccharide, because it can be glycogen, or the cell wall, which is peptidoglycan, or the protein or the enzymes which are present in it, or the phospholipid bilayer will be digested, or the glycogen reserves in the bacteria will be digested, or the DNA will be digested into nucleotides. So basically what has to happen is all the bacteria has to be digested. The word digestion should be very clear to everybody. Digestion means large insoluble to small soluble, as we study in O levels. And then ultimately the proteins are digested to amino acids, the lipids are digested to fatty acids and glycerol, the nucleic acids are digested to nucleotides, so to form useful soluble products, which of course can then be reused, like for instance when we are digesting worn out organelles, not in this case so much, but in the case of worn out organelles, the cell will then reuse all those monosaccharides or the amino acids or the fatty acids and the glycerol to form useful, the word is useful soluble products. So you need to understand, why am I writing all this in the mark scheme? This was only a 2-mark question, but you must get those 2 marks. Question 2. Glycogen and cellulose are polymers. We all know that is biological molecule chapter. Figure 2.1 shows small representative regions of a glycogen molecule and a cellulose molecule. You can see here, this is the 1,4 glycosidic bond and this is the 1,6 glycosidic bond in glycogen. So it has 1,4 and 1,6 bond. In cellulose, they're all 1,4 beta glycosidic bonds. Of course, there's no such thing as beta glycosidic, but you know, in cellulose, we have beta glucose. In glycogen, we have alpha glucose. And in this, it flips 180. You see, this 6 is down here. This 6 is up here. Why? Because it flips 180 degree because of the orientation of the beta glucose, the carbon 1. So alpha below, beta above. The OH is above on carbon 1. Please go through the chapter and understand this point of the syllabus. Describe three ways visible in figure 2.1 in which the molecules of glycogen differ from the molecules of cellulose. Now, the first and the most important thing is glycogen is branched, cellulose is unbranched. You can see it here. This is the branching. So, glycogen is branched while this is all got straight, it's unbranched. There's no branching. Then monomer is made up of alpha glucose, cellulose is made up of beta glucose. And then in glycogen, there's 1,4 and 1,6 glycosidic bonds. In cellulose, there are only 1,4 glycosidic bonds. And the last point is alternate monomer. The is not rotated 180 degrees in glycogen. It is rotated 180 degrees in cellulose. So you can see the mark scheme. And now I have sort of done it like this. I think you all can also do this. Just write the heading glycogen and cellulose. And then you just draw a line in the center and you can say branched, unbranched. So this is 1 mark. This is the second mark, alpha glucose, beta glucose. Then third mark is 1,4, 1,6 and only 1,4. So opposite, I'm writing comparing the glycogen and cellulose. Then alternate glucose is not rotated in glycogen. In cellulose, it is rotated. So you can do this. You can even write it the other way, normal way, just in a sentence. You can do that as well. Coming to the B part of the question, glycogen is found in the form of granules in mammalian liver and muscle cells. Figure 2.2 is a diagram of part of a molecule of glycogen isolated from a glycogen granule. Explain how the structure of glycogen is related to its function in cells. It's a very direct question. It's in the syllabus. This point, glycogen is a store of energy. Many branching points, so easy, faster addition, release of glucose. So they can be added and they can be removed as well. And compact, so stores much glucose in a small space. There is a large insoluble molecule, not a large, but an insoluble molecule. Does not sort of lower or change the water potential, and prevents loss of glucose from the cell, inactive, so does not take part in cell metabolism. So this was only for 3 marks. So how the structure of glycogen? And that's a very typical question which I always, when I'm teaching you this, I also say is compact, insoluble, and easily hydrolyzed. And because it's insoluble, it will not affect the water potential. So you can see how I worded it. Glycogen is a store of energy. Please do not say produces energy. Produces energy is wrong. It's rubbish. I mean, you can't write this in O level or in A levels. You know the law of thermodynamics. Energy cannot be created or destroyed. And a large number of students wrote that because they reject. Branching points easy to add and release glucose. You see, this is a very nice thing. This can be easily removed. This glucose which is right at the end. There are lots of these at the end, so we can just remove one each, and then we have a lot of glucose entering the blood cell. And it's also easy to add another one to it if we need to add some more. So idea that many branching points for easy, faster addition and release of glucose. Compact, so stores much glucose in less space. Anything compact means that you're storing it, there's less of a spread of the things. Like you have a little box with all your pencil case and everything. Otherwise, the pencil is lying here and a pen there, and all it can be spread all over your desk if you're a messy sort of a person. Compact, so stores much glucose in a less space. Then insoluble, which is very important, because it does not change the water potential of the cell. So that makes it all very easy. Of course, there's another point also which says prevents loss of glucose from the cell, because this whole molecule is an inactive, so does not take part in cell metabolism. But I think any of these three would have got you your 3 marks. Then you come to the C part of the question. Explain how the arrangement of cellulose molecules in plant cell walls is related to their functions. A very direct question, nothing very difficult, very, very direct question. Now, how is the cell wall? It's unbranched, straight chain. Cellulose molecules are arranged in parallel. Large number of OH groups projecting in all directions, so allows hydrogen bonds to form with water. Many hydrogen bonds between cellulose molecules. Cellulose molecules form microfibrils. And the last point is hydrogen bonds give high tensile strength to microfibrils. So all this is important because the hydrogen bonds will help the cell wall to resist turgor pressure. So the arrangement of the cellulose molecules, 3 marks. Unbranched, arranged in parallel rows, large number of OH groups projecting in all directions, allows hydrogen bonds to form with water, many hydrogen bonds between cellulose molecules, many cellulose molecules form microfibrils, and hydrogen bonds give high tensile strength to microfibrils, or you can even say cellulose fibers, which is of course makes up the cell wall. So unbranched cellulose molecules arranged in parallel, large number of OH groups projecting in all directions, allows hydrogen bonds to form with water, many hydrogen bonds between cellulose molecules. Cellulose molecule form microfibrils. Hydrogen bonds give high tensile strength to microfibrils. Question 3, figure 3.2 shows fruits, grapes of the grapevine, Vitis vinifera. Sucrose is transported in the phloem of the grapevine to the fruits. In the fruits, sucrose is hydrolyzed by the enzyme sucrase, which is found in cell walls. The glucose and fructose produced by the hydrolysis of sucrase pass through membrane proteins known as hexose transporters into the cytoplasm of the fruit cells. State why membrane proteins are required for the movement of molecules such as glucose across cell surface membrane. Very easy. It's water soluble. It cannot pass the hydrophobic part of the cell membrane. So if anything which is polar or water soluble or hydrophilic, it cannot pass through the hydrophobic core of the cell surface membrane. polar, or you can say water soluble, cannot pass through the phospholipid bilayer. Researchers investigated one type of exhaust transporter known as VVHT1, which is found in the fruit cells of V. vinifera. They used a mutant strain of yeast that has very few exhaust transporters in the cell membrane to investigate the properties of VVHT1. The researchers inserted molecules of VVHT1 into the cell membrane of the mutant strains of the yeast. So they took a yeast cell, which has a cell wall and then a cell membrane, and then what did they do? They inserted the molecules of VVHT1 into the cell membrane of the mutant strain of yeast. Equal volumes of mutant yeast cells with VVHT1 were kept in 8 different concentrations of glucose solution. The rate of uptake of glucose by the yeast cell in each solution was determined. All the solutions were kept at the same temperature and pH. The results are shown in figure 3.2. Here on the y-axis you have the rate of uptake of glucose in micromoles per minute per gram and concentration of glucose. So you can see how what is happening. The rate of uptake is increasing till about here and then it's leveling off. So the rate of uptake is increasing as the constant of glucose is constant. But after you've increased it from 150 to 200, a time comes when it is going to the rate of uptake is going to level off. That means the rate of uptake has become constant. It doesn't mean rate of uptake has stopped. It means, you see, here the rate of uptake is increasing. Here it is increasing. Here it is increasing. Here it is increasing. But here you see it has become constant now. So rate of uptake has become constant. Understand the question. B part 1, the researchers concluded that VVHT1 is responsible for the facilitated diffusion of glucose into the cells. Explain how the results in figure 3.2 provide evidence to support this conclusion. You see the rate of uptake when it becomes constant, what does that mean? And that becomes constant at high concentrations above 150 micromoles per dm cube. The rate becomes constant. Why? Because all the protein transporters are working at the highest rate. They're saturated. It's just like a room has a sort of a cinema hall has 10 doors. So 10 people can come in at one time. But if only 2 people are entering, that means the other 8 doors are still vacant. Only 2 people are coming, so only the 2 of the doors are being used. So what you have to understand is that around the cell membrane, there are channel proteins. It depends how many of those channel proteins are present. Now, in the beginning, only maybe one channel protein was being used, then a second was being used, then this was being, a time comes when all of them are being used. So that is why the rate increases and then it becomes constant. Because all the channel proteins, all the protein or the transporter proteins are working, or you can say they are saturated, or you can say the number of protein carriers is the limiting factor. So rate of uptake becomes constant, or you can say at high concentrations above 150 micromole dm cube, the rate has become constant. So 1 mark for that. And the second mark would be the protein transporters or the VVHT1s are working at the highest rate, or you can say they're saturated, or you can say the number of protein carriers is the limiting factor. Then coming to part 2 of the question, these researchers thought that grapevines could be modified to have more exhaust transporters to increase the size and quality of the grapes. Grapes are sweet, you know that. Size, of course, means more, bigger ones. So explain why increasing the number of exhaust transporters could be commercially important to growers of grapevines. Number 1, grapes will be sweeter, have improved taste. Concentration of sugar in the grape determines the uptake of water by osmosis. More energy for growth of the grapes, larger yield, increased income for farmers, and producing grapes in a shorter time. In a shorter time, they'll grow to a large size, and you can harvest them quickly and then sell them in the market and make a lot of money and be richy rich. Grapes will be sweeter. Concentration of sugar in grapes determines uptake of water by osmosis, more energy for growth of grapes, larger yield, increased income for farmers. Now coming to the C part of the question, figure 3.3 is a diagram of a protein in the cell surface membrane of a macrophage from a mouse. Macrophages use these proteins in antigen presentation. Non-self antigens bind to the proteins and are involved in the activation of specific T lymphocytes during the immune response. Cytoplasm, antigen binding region. State what is meant by non-self antigen. A glycoprotein that stimulates an immune response or that stimulates the production of antibodies or causes an activation of lymphocytes. So it's a foreign protein that stimulates an immune response. Like, people are allergic to pollen, so it's a foreign protein and results in an exaggerated immune response. What is meant by a non-self antigen? A foreign protein that stimulates an immune response or it results in the production of antibodies or causes activation of lymphocytes. Part 2, last part of this question. Some pathogens enter human cells. Macrophages partially digest these pathogens and present antigens to T lymphocytes during an immune response. With reference to figure 3.3, explain how T lymphocytes respond to infection by a specific type of pathogen. T lymphocytes have receptors complementary to an antigen. T lymphocytes bind to antigen. T lymphocytes divide many times. That's called clonal expansion. T helper cells secrete cytokines. Activation of B lymphocytes. T killer cells bind to infected cells and destroy them. And the T killer cells release perforin and production of memory cells of all the T lymphocytes, the T helper cells or the T killer cells. Now you can see there are lots of mark scheme points, but you could have written any four of them. T cells have receptors complementary to an antigen. T cells bind to antigen. T cells undergo clonal expansion. T helper cells secrete cytokines. Cytokines activate B lymphocytes. T killer cells bind to infected cells and destroy them. Perforin punches holes into infected cells and production of memory cells, whether it was T lymphocyte or the killer cell or the helper cell, all of them. So because you see the question was very vague, it says explain how T lymphocytes. So if you've studied this chapter and you know it well, you would understand the killer T cells and the helper T cells. So these are the ones that we're going to be talking about, and you would be, of course, if you've revised it from the videos on this chapter, I'm sure you can understand what I'm saying. So that completes this video, and we will continue the rest of the questions in the second video. Thank you very much.